Chromosomal Aberrations. UGC-Net Anthropology. Welcome to AnthroNet Academy, a study aligned with the UGC-Net Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Understanding Chromosomal Aberrations, a two-part series. To make this important topic clear and easy to grasp, we're breaking it down into two focused parts. Part 1 will explore numerical chromosomal aberrations, where the number of chromosomes changes, leading to conditions like Down syndrome, Turner syndrome, and more. Part 2 will dive into structural chromosomal aberrations, where the structure of chromosomes gets altered, including deletions, duplications, inversions, and translocations. Stay tuned and follow along. This breakdown will help you master the concepts step by step. Definition. Chromosomal aberrations, chromosomal mutations, can occur due to alterations in chromosomes' structure or number. The structural variations include deletions, duplications, translocations, and inversions, while changes in chromosome numbers are detected as aneuploidy or polyploidy. Classification of chromosomal aberrations. Category. Type. Description. Example. 1. Numerical aberrations. Trisomy. Change in number of one or few chromosomes. Trisomy 21, Down syndrome. Monosomy. Loss of a single chromosome, 2n1. Turner syndrome, 45X. Trisomy. Gain of a single chromosome, 2n+1. Patau syndrome, trisomy 13. Polyploidy. Increase in complete sets of chromosomes, 3n, 4n, etc. Triploidy, 3n, tetraploidy, 4n. 2. Structural aberrations. Deletion. Loss of a chromosome segment. Cri-du-Chat syndrome. Duplication. Repetition of a chromosome segment. Charcot-Marie-Tooth disease. Inversion. A chromosome segment is reversed end to end. Paracentric or paracentric inversions. Translocation. Segment of one chromosome transferred to another. Philadelphia chromosome, CML. Reciprocal. Mutual exchange of segments between non-homologous chromosomes. Burkitt's lymphoma. Robertsonian. Fusion of two acrocentric chromosomes at centromere. Familial Down syndrome. Ring chromosome. A chromosome forms a ring due to deletion of terminal ends. Ring chromosome 14. Acechromosome. Arms of chromosome are mirror images. Acechromosome X. Numerical aberrations. Variation in chromosome number ranges from the addition or loss of one or more chromosomes to an increase in the number of haploid sets of chromosomes. The former results in aneuploidy, whereas the latter condition generates polyploidy. Aneuploidy. Aneuploidy is a kind of numerical chromosomal aberration in which the total number of chromosomes is not an exact multiple of the haploid set. Some of the common types of aneuploidy encountered in diploid organisms are monosomy, trisomy, tetrasomy, and nullisomy. These individuals suffer from specific genetic imbalances. Nullissimy. It is the loss of both members of a homologous pair of chromosomes. It is represented as 2n2, where n refers to the haploid number of chromosomes. In humans, a nullisomic will have 44 instead of 46 chromosomes. Monosomy. It is due to the loss of a single chromosome, 2n1. Most human monosomies do not survive except females with a missing X chromosome, as in Turner syndrome, 45XO. Trisomy. It is the gain of a single chromosome, represented as 2n plus 1. The gain of a chromosome means that there are 3 homologous copies of one chromosome. In humans, both autosomal and sex. Chromosome trisomies have been reported, with variable survival rates. Examples of viable trisomies include primary Down syndrome, 47, plus 21, Edwards syndrome, 47, plus 18, and Klinefelter syndrome, 47, XXY. Many trisomies are fertile and the extra chromosome forms a trivalent at meiosis. Tetrasomy. It is the gain of 2 homologous chromosomes, 2n plus 2. A human tetrasomic zygote has 48 chromosomes. In tetrasomy, there will be 4 homologous copies of a particular chromosome. Why aneuploidy occurs? The major cause of aneuploidy is non-disjunction, failure to separate, of homologous chromosomes or sister chromatids during meiosis or mitosis, respectively. It occurs spontaneously due to chance failure of the cellular machinery. This results in gametes or cells with abnormal chromosome constitution. Polyploidy. An organism with multiples of the monoploid number of chromosomes is euploid. A euploid organism with 3 or more complete sets of chromosomes is polyploid, and those with 2 sets are diploid. They may have odd, triploids, or even number, tetraploid, hexaploid, of chromosome sets. Most species of animals are diploid, but they sometimes produce tissues that are polyploid. For example, human liver exhibits varying degree of polyploidy, endopolyploidy. Polyploidy is fairly common in plants and has played a significant role in speciation, particularly allopolyploids. Types of polyploids. Polyploids are classified into auto and allopolyploids depending on whether the chromosome sets are derived from a single or different, closely related, species, respectively. Examples of autopolyploid include seedless bananas and watermelons, and those of allopolyploids are triticale, wheat and rye, and bread wheat, natural hexaploid. Mosaicism and chimerism. Mosaicism and chimerism refers to an organism with 2 or more genetically distinct populations of cells, interorganismal genetic heterogeneity. The term mosaicism is reserved for individuals with genetically distinct cell lines that originated from a single fertilized egg, while the chimerism refers to those who originated from more than one zygotic fusion, dispermic chimera. Mosaicism example. Mosaicism also accounts for the variable expressivity of certain forms of syndromes such as Klinefelter syndrome, Turner syndrome, Klippel-Trenaunay syndrome, Down syndrome, and Pallister-Killian syndrome. Chimeras can be produced in a variety of ways, such as fertilization of 2 separate eggs by 2 sperms, dispermic chimera, produces a tetragametic individual. The different tissues of the organism are made up from cells derived from one or both zygotes. Microchimeras are produced when fetal stem cells or maternal cells cross the placenta. Fetal eternal microchimerism, or following blood transfusion, transfusion-associated microchimerism, or organ transplantation or twin chimeras, through connections between the placentas of dizygotic twins. In all these cases, two zygotes are not fused but exchange of cells and genetic material occur. Uniparental disomy. Normally, the two chromosomes of a homologous pair are inherited from different parents, one from the father and one from the mother. But sometimes both chromosomes are inherited from the same parent, a condition termed uniparental disomy. Many times a trisomic embryo loses one of the triplicate chromosomes early in development, and if by chance the two remaining chromosomes are from the same parent, it results in uniparental disomy. Aneuploidy in humans. Syndrome Down. Aneuploidy trisomy 21. Frequency, live births, 1 in 750. Symptoms: low IQ, 20 to 50, short and broad hands, broad flat face, slanting eyes. Dental abnormalities, wrinkled tongue, small land-arched palate. Remarks: incidence increases with mother's age, may survive to adulthood, in some it is a familial trait. Syndrome Turner. Aneuploidy monosomy, XO. Frequency, live births, 1 in 2500. Symptoms: phenotypically females, most are sterile, short stature, broad chest, rudimentary ovaries, no menstruation, poor breast development, hearing loss. Remarks: many females are somatic mosaics, most of them suffer from nonverbal learning disabilities, NLD. Syndrome Klinefelter. Aneuploidy trisomy, XXY. Frequency, live births, 1 in 500-800. Symptoms: sterile males, tall, mildly impaired IQ, small testes, gynecomastia in some. Poor beard growth. Remarks: most common sex chromosome disorder. Syndrome Edwards. Aneuploidy trisomy 18. Frequency, live births, 1 in 60-100, 8,000. Symptoms: suffer from multiple congenital defects involving almost all organs, mental retardation, poor survival, most die within 6 months. Remarks: majority are females, prevalence increases with maternal age. Syndrome Patau. Aneuploidy trisomy 13. Frequency, live births, 1 in 50-100, 12,000. Symptoms: polydactyly, unusually small eyes, cleft lips and palate, congenital heart and kidney defects, feeding difficulties, die within 3 months. Remarks: incidence increases with mother's age. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.